Welcome to Dyadic's investor presentation. 2021 is setting up to be a breakthrough year for Dyadic, for our C1 technology platforms, adoption and use in animal health and human health, infectious disease, and a variety of other applications. Additional risks can be found on our website at www.dyadic.com. As we enter 2021, we have a solid financial position, strong insider ownership, but our greatest asset is the C1 gene expression platform, which has the capabilities and the potential to revolutionize the development and production of biologic vaccines, drugs, and other biologic products. In a relatively short time from our transition from industrial biotech to biopharmaceuticals, Dyadic and the C1 gene expression platform have started to embed itself in animal and human health as well as infectious disease. We've demonstrated through scientific data, which is growing and expanding, that we can produce things quickly in larger amounts at lower cost. We've done so by advancing science, modifying the technology, continuing to make it faster, more robust, more versatile in the applications that it can be addressed and used for. We've done so with guidance from some of the strongest management team members that are highly motivated, along with very experienced board members in biopharmaceuticals. Our management team and board of directors are focused on creating shareholder value. We have deep industry experience, both as a management team and a board of directors. As you'll note from the slide here, Dr. Bose comes from Pfizer, 34 years of biologic manufacturing, mainly antibodies and other therapeutic proteins. Perry Buckland comes from Merck, 29 years, where he spent in biomanufacturing, mainly in vaccines. Michael Tarnock has broad business development and corporate governance strengths coming from Pfizer, where he was there for over 3 decades. The management team, as you can see, Dr. Chellitt, Matt Jones, and Ping Rawson all have the relevant experience to oversee the business development efforts, the science, and the management of our resources. You can see from this slide that the company history is a solid history of growing a portfolio of high-value collaborations. The history in the industrial biotech sector shows you that we licensed the technology to some of the largest companies in industrial biotech, from Abengoa Bioenergy, BASF, and Codexis and Shell Oil, are just to name a few, where we raised over $30 million of non-dilutive, non-exclusive license agreements. We then sold the industrial biotech business to DuPont for $75 million. We kept the rights to human and animal health from the pharmaceutical, where we have the exclusive sub-licensing rights to use C1 for human and animal pharmaceutical applications. We've engaged with leading biotech companies, pharmaceutical companies, and governmental agencies on a global basis. And we now have the ability, if we choose, to re-enter the industrial enzyme business, as our 5-year non-compete with DuPont ended on 12/31/2020. As you can see from this slide, we've had significant advances in the science and in our business development efforts. On the human health side, we entered into more than a half a dozen fully funded collaborations with top-tier pharmaceutical companies, top 5, top 25, and China's largest pharmaceutical company by market cap. In the animal health sector, we entered into additional funded collaborations with top-tier, top 4 animal health companies, so that we've now entered into collaboration with all 4 of the top 4 animal health companies. In the research and license part of our business, on the non-exclusive side, we entered into a collaboration with WuXi Biologics, expanding our presence in Asia. We extended the collaboration with an affiliate of a top-tier pharmaceutical company. On the COVID-19 initiatives, we were able to demonstrate the power, the productivity, and the speed that we can develop stable cell lines to produce vaccines for pandemics and other biological outbreaks. We entered into a research agreement with the Frederick National Laboratory, where they gave us their gene sequences, which we've now turned into vaccine candidates and returned to them for their evaluation. We expanded our relationship with the IIBR. We developed a vaccine candidate with them moving towards potential preclinical final stages towards maybe a clinical trial in 2021. We also have worked with the ZAPI, the Zoonosis Anticipation Preparative Initiative, since 2015. We achieved significant results in animal health, but we've also worked with 3 of the top 20 coronavirus scientists that were within ZAPI to develop vaccine candidates that we're now moving through hamster trials that started last month. In addition, we've advanced the science. It's more robust, it's more versatile, it's more productive. It produces proteins more stably. We look at 2021 as a significant milestone year to turn some of these opportunities into license agreements and to expand into human health, improve the safety and efficacy of 1 or more vaccine candidates from C1. The C1 technology platform is aligned with global trends. The pharmaceutical industry is in great need for a more efficient cell line that produces more for less. Dyadic C1 expression platform provides a novel solution to infectious diseases, including pandemics. It brings new biologics to the market faster and allows for broader access and affordability to healthcare for patients globally. We have a growing aging population, and we have a growing need to be able to address pandemics and epidemics faster and more reliably. We believe C1 helps to answer those challenges. As this slide highlights, having the ability to produce biologic vaccines and drugs faster and larger quantities at lower cost provide enormous opportunities for us to leverage our C1 technology in collaboration with animal and human health pharmaceutical companies and governmental agencies on a global basis. These markets are in the hundreds of billions of dollars. If we can just address a small fraction of those with the C1 technology platform, we can help make healthcare more accessible and affordable to patients globally and bring a significant value to Dyadic shareholders along the way. Now I'd like to highlight why C1 has competitive advantages over other cell lines to help disrupt the biological manufacturing of vaccines and drugs. C1 is a hyperproductive cell line. It has unique morphology that leads to low cost, high purity and yield, and scalable benefits for the manufacturing of proteins. C1 was initially engineered to produce enzymes for the textile industry, where we spent over 2 decades producing enzymes at extremely large scale. Proven low cost, high yield, high purity, robust technology. C1 genome is fully sequenced and annotated, and we have a full set of genetic tools to program the cells with genes of interest for both animal and human health and infectious disease, as well as other growing biologic product opportunities. C1 received the GRAS status, generally recognized as safe, from the US FDA, and we're now on path to move into a potential phase 1 clinical trial with a C1 vaccine candidate for SARS-CoV-2 that will additionally add greater safety and efficacy data to help support the growth, adoption, and use of C1 on a much wider basis in human pharmaceuticals. For animal pharmaceuticals, we've already proven its safety and efficacy in mice, in cattle, and other animals are on the way in the ZAPI program and otherwise with some of our collaborators. Again, it's really all about the cell line, its hyperproductivity, its purity, its robustness, its versatility, the speed of being able to develop stable cell lines quickly. They can produce a lot in a short amount of time for less. For example, not only in vaccines, but in monoclonal antibodies, where we can actually produce up to 3 to 4 batches of antibodies in the same time frame that it takes a CHO cell to produce 1 batch. And we can do these tech transfers very quickly and very rapidly in standard stainless steel or single-use bioreactors. C1 produces monoclonal antibodies that have similar activities to those produced from CHO cells. The difference is the C1 platform has superior production potential versus Chinese hamster ovary cells or CHO cells. C1 grows antibodies faster and higher yields at lower cost. C1 can make a huge difference in bringing therapeutic treatments for COVID-19, rheumatoid arthritis, cancer, or a growing number of other cures for disease that monoclonal antibodies or bispecific antibodies or multispecific antibodies are addressing. This slide lays out a subset of the broad set of biologic vaccines and therapeutic proteins that can be potentially produced in larger quantities at lower cost more affordably than Chinese hamster ovary cells or other cell lines. To give you two examples, monoclonal antibodies on average produce 4 grams per liter in 12 to 14 days. Here we demonstrated in an antibody expressed by C1 at 24.5 grams per liter in 7 days. Antigens, if you recall from earlier presentations, in the ZAPI program, we demonstrated the SPV antigen at 1,800 milligrams per liter. That was compared to 6 milligrams per liter of the same antigen produced from baculovirus, or 300 times less productivity than C1. In this case, with our SARS-CoV-2 RBD antigen vaccine candidate, we've demonstrated over 3,500 milligrams per liter in just 5 days. This just shows you the potential power and the breadth, the scope, and the depth of the applications and the value that we can create for biopharmaceutical industry and to make healthcare more accessible and affordable to patients on a global basis. At Dyadic, we have our eyes on the prize. The prize is to bring value to our customers that'll lead to lower healthcare costs on a global basis that'll create significant value for Dyadic shareholders. Our business development efforts are focused on hundreds of billions of dollars of opportunities of where we can apply our C1 technology platform. With Big Pharma, we're receiving fully funded proof of concept collaborations for specific therapeutic agents. That brings us with the potential for upfront access or license fees, milestones, and royalty payments. That also allows us to advance the C1 technology platform using their dollars as we learn how to modify C1 to meet their needs. For smaller biotechs, we've been getting either equity upfront or we get fully funded research development, milestones, and royalty payments. We're in addition to that developing the technology platform and advancing it and broadening its use in applications by glycoengineering the cell lines to make human-like glycan proteins, deleting proteases to make more stable, higher productive proteins, and potentially moving biosimilars or biobetters along the way, and including potential vaccine candidates for COVID-19 and or monoclonal antibodies in the multiple programs we have ongoing on a global basis. The C1 gene expression platform is helping to overcome gene expression and production challenges in animal health and human therapeutics and human vaccines. Applying C1 in animal health allows the animal health companies to produce vaccines and drugs for companion animals and farm animals quicker, faster, and cheaper. For human therapeutics, it's about better economics, the potential to serve a broader patient population more affordably, and the ability to overcome pre-existing gene expression bottlenecks and production challenges with other cell lines. For human vaccines, C1 novel recombinant vaccine proteins have been produced at higher yields, at lower costs, more stably. It's well suited to the scales, costs, and timelines required for vaccine manufacture, including for use in pandemics. Dyadic and C1's involvement in the Zoonosis Anticipation Preparative Initiative, ZAPI, since 2015 has provided Dyadic with the expertise and the funding to help advance the C1 platform for hyperproductivity of antigens produced from C1. In ZAPI, C1 was selected for the production of ZAPI antigens after a competitive process. To give you an example, in ZAPI, we were able to show that you could produce 300 times greater productivity of the SPV antigen than that was produced from baculovirus cells. In ZAPI, we also were able to demonstrate the safety and efficacy of the C1-produced SPV antigen in cattle. Additional animal studies are planned for 2021 with C1-produced SPV and RVFE antigens. Dyadic's collaboration with the Israel Institute for Biological Research, or the IIBR, is another example of a successful execution of a government-sponsored program. This collaboration initially led to the production of an enzyme produced from C1 as a countermeasure against nerve agents such as sarin and VX gas. The success of that proof of concept experiment led to the decision by the IIBR in February of 2020 to expand its collaboration with Dyadic to use C1 to develop a potential vaccine candidate to help combat the COVID-19 outbreak. In collaboration with the IIBR, we've developed a C1 RBD-CoV-2 strain that has the potential to be produced in very large volumes at very low cost with, according to the mice studies, high titers in neutralizing antibodies. Currently, the IIBR is conducting a challenge study using transgenic mice expressing a human ACE2, which will be infected with the COVID-2 virus. As you can tell, there's almost an endless amount of opportunities of where one can apply the C1 technology platform for animal health, human health, infectious disease, and other biological products. The COVID-19 pandemic shined the spotlight on the need for rapid, safe, and scalable vaccine and antibody manufacturing platforms. We and a growing number of scientists and scientific institutions and governmental agencies believe that C1 has the potential to meet that need. To develop an affordable, scalable, safe, and protective vaccine and antibody production platform to combat infectious and other diseases such as COVID-19 and allow for fair and equitable access around the globe. The data tells a story. Just like in Zappy, when we made 300 times more of the SPV antigen than from baculovirus cells, C1 has done it again. For the COVID-19 pandemic, we've been able to produce very rapidly a stable cell line that initially could produce 1 gram per liter in 5 days. We've now optimized that, and we can produce up to 3 grams per liter in 4 to 5 days. This correlates to the potential to produce billions of doses of that vaccine candidate monthly, not yearly, at lower cost that can be tech transferred flexibly all over the globe. Based on multiple assays and animal studies, it's been demonstrated that the binding kinetics and the neutralizing properties of the C1 expressed SARS-CoV-2 RBD antigen are virtually identical to that as the RBD antigen expressed from CHO cells. The mice studies confirmed that the RBD antigen produced by C1 created neutralizing antibodies against the SARS-CoV-2 in mice. Additional animal studies are underway to assess the immunogenicity and protective efficacy upon challenge in hamsters and transgenic mice. Currently, there are 9 C1 SARS-CoV-2 vaccine and monoclonal antibody programs ongoing. In addition to these 9 programs, we're in discussions with numerous other parties interested in using C1 to express potential vaccine and antibody candidates of their choosing. The model protein that we're using in our glycoengineering program of C1 is nivolumab. Nivolumab could be a potential biobetter, biosimilar to Opdivo, which is an $8 billion drug for Bristol Myers Squibb. The program is designed not only to create a pipeline product opportunity to out-license that technology of nivolumab, but to demonstrate that we can make monoclonal antibodies with human-like glycan structures that perform virtually identical to the monoclonal antibodies produced from CHO cells. As we mentioned earlier, there's over $200 billion worth of monoclonal antibodies, and it's one of the fastest growing sectors of the biopharmaceutical industry. So if we're successful in producing nivolumab, the adoption and use of C1 to make monoclonal antibodies will accelerate at a rapid pace. We're working with the University of Oslo to try to demonstrate and determine the viability of C1 to express influenza virus antigen proteins. Previously, C1 was tested by Sanofi Pasteur in an influenza vaccine trial versus baculovirus. In that trial, C1 showed that the hemagglutinin was safe and well tolerated in mice, and that C1 produced hemagglutinin was at least as immunogenic in mice as the baculovirus hemagglutinin. As you can see by this slide, the statement of at least immunogenic in mice is understated, as we used at 10 units per gram, 1/3 the amount of the protein and got a better result and made it for far less. I hope you take away from this presentation the possibilities that C1 has the opportunity to disrupt biologic vaccine and drug manufacturing. We have the potential to bring biologic vaccines and drugs to market faster in greater volumes at lower cost. Compared with other pharmaceutical expression systems, C1 offers significant competitive advantages. Our emphasis is on large and growing addressable human and animal health markets, including infectious disease. We're well financed, no debt, and we're complemented by partner funding on ongoing R&D collaborations that help us advance our science, make it more broadly applicable and more productive and more powerful to apply to virtually an endless number of opportunities. I want to thank you for attending my presentation and learning about our quest to make healthcare more accessible and affordable to patients globally for traditional biologic vaccines and drugs, and help combat pandemics such as COVID-19 and any future pandemic the world may face.